Here I have some tryptic soy agar that I made. It's a media to grow bacteria. Could have also molds and fungi on them. This is completely sterile. And so what we want to do is grow single colonies of bacteria on them. And I'll show you an example. This is a plate with one type of bacteria on it. You start streaking, and this is loads and loads and loads of bacteria, but then you kind of sterilize it, get less and less, and then you get individual tiny little colonies right here. So each little colony grew from one bacteria. So how we are doing it is like what our book says, and this is how you plate according to our lab manual. One day you may streak it a lot differently than this, but it's all basically the same, where you streak one quadrant, sterilize your loop, streak the next, sterilize your loop, streak the next, sterilize and streak the next. That way you get these individual colonies on here. And then you can use individual colonies to streak another plate or to do a gram stain or whatever it is you're planning on doing. So if you'd like to practice, you could just draw a plus sign in here, and it might be easier for you to gauge your streaking, or you can practice on a piece of paper just drawing it out and before you actually do your work. So right now these plates are upside down, and that's the easiest way to handle them, is put the cover on the bottom and grab the bottom of the plate. Lift, do your work, put it back down, and put it back down. Easiest way than just taking this off and then trying to grab this and then, okay, then go grab your cover and putting it back on nicely. So much easier to just flip them over. And you're also going to be incubating them like this, upside down, because some of these could have some condensation in them. If you incubate them with the condensed side up, this will drip onto your plate and your bacteria will just smear all over the place. So they will be incubated upside down too. Alright, so we're going to label our plate. It is kind of labeled TSA, tryptic soy agar. I am going to label with the date. So today is the 9th of August. Initials, or identifying it that it's your plate, so that's my initials. I also put on what is going on it. So I am going to put in colony number one, and I believe that's all I need. So this is our loop, and you kind of see it's kind of got a ring on the end, so hence the name loop. This is an incinerator. This is what we use to sterilize our loop. You can see it's nice and hot, so don't touch. On and off switch if it's not on. So we're going to hold it at this angle, sort of like a writing pencil. Alright, I am going to streak from my colony number 2, and that is my plate right there. And here is my plate all labeled, ready to go. First thing I must do, sterilize my loop. 2 to 4 seconds. 1, 2, 3, 4. We have to let it cool, or else we're going to kill our bacteria, or we're going to stab this plate and melt it. So we have to let it cool for about 10 seconds. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now I'm going to take a colony from my plate. I'm going to take a colony that is all by itself, because that is one bacteria and only one grew this colony. So I'm going to take from this one, put it back down. I'm going to grab my plate, and I streak in the upper outer corner when I streak. So I'm going to get my bacteria on there, and I'm going to keep streaking through this whole quarter of the plate. That's done. I'm going to sterilize. 1, 2, 3, 4. Cool. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now I'm going to lift my plate, and I have my streak in the upper outer corner from me. I'm going to go to the top, go back in here, and to our second quadrant about 3 or 4 times. And then I'm going to streak downward for my second quadrant until that quadrant is full. I'm going to put it back down. 1, 2, 3, 4 to sterilize. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 to cool. I'm going to lift my plate up. I had turned it around a little bit, and here's my second quadrant. I'm going to take bacteria from the second quadrant in the upper corner and go streak across to the third quadrant, and then I'm going to go down without touching that second quadrant again until this third quadrant is done. Sterilize. 1, 2, 3, 4, 5. It was supposed to be 4. Cool. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. Grab my plate so that my third quadrant is on the upper outer away from me. Then I put my loop into it and streak into the fourth 2 or 3 times, and then streak downward to fill that fourth quadrant. Then you got to remember, sterilize your loop afterwards, because you do not want to put bacteria into your drawer where the loop is kept.